In this lecture, we'll examine osmoregulation and excretion. This is chapter 44 in your book. So one example is the wandering albatross. This is a kind of seabird that lives on the sea day and night, pretty much year-round, only reproducing on land. And one question is, how does it survive in such a salty environment? It would, of course, only have seawater to drink, and it would only have fish to eat, which have very salty flesh. So one overriding concept in this particular chapter is the idea that we have to have a balance between the various salts in our body and the amount of water. And this idea really comes back from this early idea that water is essential for life. So Darwin, for example, in a letter to his colleague, spoke of a warm little pond in which the first molecules might have formed that could have led to life. And since then, a number of people have further done research along those lines, and now one of the current theories is perhaps life arose at hot springs. You can, of course, see in this picture hot springs in Japan that are being enjoyed by monkeys. So in a sense, you yourself, you are a kind of a pond. You have cells that are themselves mostly water. So for example, the average human adult male is approximately 60% water. Females are similar also, they're around 60%. The actual percentage varies a little bit with the fat content of the body. But you're mostly water, so your cells are mostly water, and they themselves are bathed in a fluid that surrounds your cells, which is called interstitial fluid. And then that interstitial fluid is in communication across the walls of leaky capillaries in your body with the blood plasma, again, which is mostly water. So controlling the levels of salts, such as sodium, chloride, potassium, calcium, magnesium, and so forth, is very important within this aqueous environment that bathes our cells. So water is this common thread in evolution, from single-celled organisms to much more complex multicellular organisms such as yourself. So since we arose from simple organisms in some sort of pond or hot spring, it's not surprising then that all our cellular processes depend on water and the exact solutes within the cells. So to keep our cells healthy, the concentration of ions in water must be kept within a narrow range. And that's essentially the main topic of this chapter. So osmoregulation is the general term for processes by which an organism controls solute concentrations and balances water gain and loss. So different animals have different challenges. So for example, a desert mammal, such as the sandy inland mouse, which is found in Australia, it has the problem that it survives or lives in a very dry environment. So it can never drink water, and it basically gets its water from seeds that it eats, and also it gets water through the breakdown of things like sugar, such as glucose, into CO2 and water. So that happens through aerobic respiration in your body also. For a desert mammal like the sandy inland mouse, this water that's generated through aerobic respiration actually becomes a significant part of the water that it generates and uses for its life. For something like a human, it's not as significant. What else? So freshwater animals survive by conserving solutes. So let's take the example of the sockeye salmon. This is an animal that can go back and forth between ocean water and freshwater. When it's in freshwater, its tissues are much more concentrated. In other words, they have a higher solute concentration than the environment, than the lake or the stream that it's in. This means that the water concentration is higher outside of the animal, and water will tend to move into the animal, into its tissues. And at the same time, the salt concentration of the cells, the interstitial fluid, the blood plasma, and so forth is higher inside of the animal compared to the environment it's in. So the salts will tend to diffuse out of the body of these sockeye salmon. So they're going to need to conserve solutes. They need to conserve their solutes because they're losing them. And they're going to need to try to prevent water from entering into their body, but also when it does, which they'll have to excrete that water. And so we'll talk about the details of that in a second. Now, the other thing, another topic of this chapter is excretion. So the term excretion is really a general term for the elimination of any material from the body. And it can be through the urine, feces, lungs. As you breathe out, for example, water vapor, and through the skin. And so you can excrete nitrogenous waste, such as ammonia, urea, uric acid. You can also excrete other things like ions, like calcium and things like that. Okay, we're going to go into more detail, but this is a general overview of what we'll be talking about. So osmoregulation is based on balancing uptake and loss of water and solutes. So let's think about water first. The driving force for the movement of water across the plasma membrane is going to be the concentration gradient for water. So water will always move from an area of high water concentration to an area of low water concentration. And likewise, the driving force for the movement of a solute across the plasma membrane is its concentration gradient. So for example, we could take sodium. If sodium is typically in a higher concentration outside of a cell than inside, so just by passive diffusion, there's a tendency for sodium to enter into cells. Now that's just by passive movement. Of course, we have transport proteins that can move things in an active way, and that's going to generally take energy in the form of ATP, or it could be energy in the form of a concentration gradient of another ion to then fuel the movement of a molecule across the cell membrane. Okay, so basically water and solutes move passively down their respective concentration gradients. Let's now take a look at an experiment. This is based on a figure from your book. I've modified it slightly. We have two solutions with different concentrations of glucose. So you can see here we have water and we have glucose, which is represented as these yellow molecules. So we start off with a situation like this. We have two separate solutions. We have a selectively permeable membrane, shown with a dotted line, and this membrane is going to only allow water to pass through it, but not glucose. So given this experiment, in which direction would you expect there to be a net movement of water? In order to analyze a set of experiment, you have to understand that the solutes, in this case glucose, actually take up physical space. And so this means there's less room for water on the left side of this beaker than on the right. Therefore, the water concentration is higher on the right than on the left. And water is just like any other molecule, will move down its concentration gradient. So in this case, water will move from right to left. It'll simply move down its concentration gradient from an area of high water concentration to an area of low water concentration. Now, if we let this experiment go on long enough, and we allow the two solutions to eventually have the same water concentration on both sides, well, this membrane is permeable, so water will continue to cross through this membrane, which is permeable, and the pores are large enough for water to pass through, but not glucose. So water will continue to move from right to left, but for every molecule that moves in that direction, there'll be one molecule that moves back in the other direction. And so there'll be equal rates of flow in both directions, and therefore there'll be no net water movement at that point. The term osmosis is simply the movement of water across a selectively permeable membrane in response to a transmembrane difference in water concentration. So this is an example of osmosis. This is osmotic movement of water from right to left. So the question is, what causes this movement of water? In this case, we've set up the experiment so it's glucose, right? We have added glucose in a higher concentration on one side than on the other side. So in a sense, it's the solute concentration that affects the water concentration and therefore drives this osmotic movement of water. So the answer then is, as I said, is a difference in solute concentration. So by having more glucose on the left side compared to the right side, we affect the concentration of water, and therefore water will move down its concentration gradient. And the reason I bring this up, and I sort of talking about this a lot, is simply because you'll often see explanations for osmosis in which they talk about water moving in response to a solute concentration. And so another description of osmosis may be that it's the movement of water across a selectively permeable membrane from an area of low solute concentration to an area of high solute concentration. And that's a perfectly valid way of describing exactly the same phenomenon, because it's correct in the sense that the solute concentration affects the water concentration and therefore affects this diffusion of water down its concentration gradient. Since this is biology, we're often interested in whether water enters or leaves a cell by osmosis. In order to determine that, we need to quantify the solute concentration on either side of the cell membrane. The way we do that is with osmolarity, which is defined as the number of moles of solute per liter of solution. Another term for this is osmotic concentration. We can use osmolarity to compare solutions. For example, if two solutions have the same osmolarity, they're said to be isoosmotic. If a solution has a lower osmolarity than another solution, it's said to be hypoosmotic relative to that other solution. If a solution has a higher osmolarity than osmolarity than another solution, it's said to be hyperosmotic. Let's give an example. So the osmolarity of human blood is about 300 milliosmoles per liter, written this way, small m, OSM, to mean osmoles, and then per liter. And seawater is 1,000 milliosmoles per liter. Therefore, the human blood is hypoosmotic relative to seawater. Seawater is hyperosmotic relative to human blood. So let's ask a question. Why is drinking seawater a bad idea? Well, we can sort of figure it out already based on just what we've discussed so far. If two solutions differ in osmolarity, the net flow of water is from the hypoosmotic or less concentrated solution to the hyperosmotic or more concentrated solution. So if we have a castaway and he gets desperate and he drinks seawater, first of all, what will happen is the salt and water will enter his blood plasma. That's going to be principally through the small intestine where you do a lot of absorption. The water will flow then. So now he's going to have essentially blood plasma that'll be have a higher salt concentration than normal, because as we just discussed, the salt concentration of seawater is about 3 times that of human blood. So this means that water will now flow from his cells into the blood plasma. So you can imagine this side being the cells, and here's his blood plasma. So water will exit his cells. This will make his cells lose water, and they'll become smaller in volume. And so that's bad. That'll really disrupt things in his body, so it could even kill him. This next slide just simply summarizes what we've been talking about. Water moves down its concentration gradient from an area of high water concentration, which is going to be an area of low solute concentration, to an area of lower water concentration, which is an area of high solute concentration. Animals can maintain water balance in one of 2 ways. One is to osmoconform. So for example, hagfish are osmoconformers. They maintain an internal osmolarity that matches seawater. So they don't really attempt to regulate their osmolarity. Their blood, interstitial fluid, and tissues, cell intracellular environment would be more or less in conformation with seawater. In other words, it'd be extremely salty. Another strategy is to osmoregulate it. So our example here of an osmoregulator is a cod. This is a type of ray-finned fish. Now, what it's going to do is expend energy to control the water uptake and loss. And in this case, a cod is a fish that lives in the ocean. Its tissues are less concentrated than that of seawater, so they're essentially in a hyperosmotic environment. But it's also possible to be an osmoregulator and be in a hypoosmotic environment. An example of that would be a fish that lives in freshwater. It's going to have more concentrated blood, interstitial fluid, and intracellular solutions compared to that of the environment. This next picture here is a sockeye salmon, and it's an osmoregulator, just like the cod. And it actually goes back and forth between the ocean and fresh water. So in a hypoosmotic environment, it's in fresh water, and it's going to have to discharge excess water. In other words, there'll be osmotic movement of water into their tissues, and they'll have to get rid of the excess water. Conversely, when they're in the ocean, their tissues are less concentrated than the seawater. So they're now in a hyperosmotic environment. And when they're in that sort of environment, water will move from the tissues into the seawater. And so they're going to have to take in water to offset this osmotic water loss. The majority of animals are stenohaline, meaning they can't tolerate substantial changes in external osmolarity. A couple classic examples of that would be goldfish. Now they live in a freshwater environment, and they're stenohaline. If you were to take a goldfish and put it in the ocean, it would die immediately. Cod are marine fish. They're also stenohaline, so they can live perfectly well in a marine environment, but if you put them in a lake or river, they would die immediately. Now, there are some animals, they're the minority, but there are urea-haline animals. These can survive even with fairly large fluctuations in external osmolarity. So, as I mentioned earlier, salmon would be a good example of a urea-haline animal. It's able to live in both, in this case, in the ocean as well as in fresh water. And the origin of these words, so Greek stenos means narrow. Urus is Greek for broad, so these are able to have a broad environment, and halanos, it means of salt. So some urea-haline means that they can have a broad range of salt for their environment, and then stenohaline would mean they have a narrow range of salt in their environment. We'll now look at marine animals. So the majority of marine animals are osmoconformers. So for example, if we look at marine invertebrates, such as a lobster, it's an osmoconformer. So it's perfectly happy being in the extremely salty environment of seawater. And they're going to simply conform. Their tissues will have very similar overall concentrations of solutes. So again, in this case, they don't have blood, but they have hemolymph, so that would be similar in concentration to that of the ocean. And their interstitial fluid, well, they don't have interstitial fluid, but the hemolymph, again, would be similar to that of the ocean. And their cells would have a similar overall ion concentration, similar to that of the ocean. Now, that's the overall value, however. So if we look at specific ions, well, for example, the hemolymph of a lobster has a much lower magnesium concentration than that of the ocean. And so it does regulate individual ions, but just the overall concentration is very similar. Okay, so many marine vertebrates and some marine invertebrates are actually osmoregulators. So marine bony fish are hypoosmotic to seawater. So here's an example of a bony fish, which is the cod, which is a ray-finned bony fish. So its blood is hypoosmotic. And here's another example. Lungfish is another type of bony fish. This is a lobe-finned fish. So it's a different group than the ray-finned fish. It's also has blood that's hypoosmotic. So these marine bony fish balance water loss by drinking large amounts of seawater. Why do they lose water? Well, it's because, remember, the environment is very salty around them. And so they're going to have a higher water concentration inside of their body compared to the external environment. And so there's going to be a tendency for water to leave their bodies. To make up for that, they're going to have to drink water, and then eliminate the ingested salt that comes with the salt water out through their gills and kidneys. And we'll see a picture of that in a moment. So just a quick break from talking about fish. Let's just use an example to illustrate this idea. So if you cut up some cucumbers and put salt in them, in which direction will there be net movement of water? Okay, the answer is that there'll be a net movement of water out of the cucumber cells and into the salty fluid around the cells. So very quickly, the salt will start to draw out water, and so you have this very salty or briny solution around the cells of these slices of the cells within these slices. And so again, there's going to be a higher water concentration inside of the cells compared to outside, and so water will move down its concentration gradient from inside the cells and into the briny solution around these cucumber slices. And so this is essentially what's happening with these marine bony fish. They have a lower salt concentration inside compared to the environment, and so there'll be water loss across various surfaces of their body. Okay, so now that we have the general concept, let's look at some more detail. Here's a cod. So we've talked about, here's water shown with the lighter blue. So they're going to lose water, right? We've established that. They're going to lose water to this very salty environment as water moves down its concentration gradient. Where are they going to lose water? Well, the major place they're going to lose water is across the gill surface. Remember, we talked about respiration. There's a very large surface area with the gills. The gills are also very delicate. There's a lot of, they're highly vascularized. They have a relatively thin, distance between the inside of the animal, which would be the blood plasma, and then the external, external environment of the animal. So that's a very difficult place to control things like water movement. And so there will, in fact, be water loss across the gills. So there's osmotic water loss to the gills and other parts of the body. Now, there also will be water loss across the skin, but of course, the skin is covered in scales. And so overall, there will be quite a bit of water loss across the overall surface of the body. But the gills are certainly a bigger area where there's more loss, simply because those structures are so very thin and delicate and so forth. Okay, so water is leaving, and then at the same time, to make up for that, how is this animal going to deal with it? Well, it's going to drink water. So it's going to drink water, shown here, and that water is very salty. Okay, so it's drinking very salty water. It's water that's actually saltier than its own tissues. So how does it deal with that? Well, it needs the water, but it doesn't really need all that salt. So what a fish has are very specialized structures within its gills that are involved in the excretion of salt ions. So it'll excrete ions such as sodium, potassium, chloride, calcium, and so forth out across the gills. At the same time as it eats food, there's going to be a gain of water and salt with the food. So again, that's good. It needs the water, but it doesn't need the salt that comes with the food. So it's going to excrete salt through the gills. So this is the main region of salt excretion. But there are also the kidneys. So fish do have kidneys, and they will excrete urine, which will have salt ions, and also a small amount of water. So it's going to attempt to make very concentrated urine. And the reason is because it's already losing a lot of water across its gills, so it really doesn't want to lose any more. And so it's very advantageous to create concentrated urine with a lot of salt in it. Okay, so far we've talked about regulation of solutes, such as sodium, chloride, potassium, magnesium, calcium. Um, and we've also talked about water balance. But there's also another problem, which is as we metabolize certain foods, such as protein or nucleic acids, those have a lot of nitrogen. And so you get the buildup of nitrogenous waste, such as ammonium and urea in the blood. Now, in us, as humans, we get rid of that through the urine. But interestingly enough, sharks have a slightly different strategy. They actually have a fairly high concentration of urea in their bodies. Oh, I should mention that urea is bad because it can denature proteins or unfold proteins. Sharks have a fairly high concentration of urea in their bodies, but they deal with this, but they have it because, it's an interesting way. They have another chemical, trimethylamine oxide, or TMAO. And this actually protects them from the denaturing effects of a nitrogenous waste such as urea. So interestingly, what sharks have is they will have relatively high concentrations within their tissues of salt. So they'll have typical salts. In addition, they'll have the urea, and in addition, they'll have this trimethylamine oxide. So when you start to combine all those, you find that the tissue in sharks is actually hyperosmotic compared to seawater. So it's slightly above the concentration of seawater. And for this reason, sharks are going to take in water by osmosis, and mostly, as you'd imagine, it's through the gills, just as in other fish. So the water will enter into their blood through the gills. They're also going to take in water in their food. And so they're essentially going to be the opposite of the fish we just talked about, like the cod, which is instead of losing water to the environment, they're actually going to they're going to have a slight excess of water coming into their bodies. And so they're going to have to get rid of that. So they're going to produce urine to get rid of that excess water that enters their body primarily through the gills by osmosis. Now, at the same time, they do take in a little bit too much of certain salts, such as sodium and chloride in their food, as well as sodium chloride that enters by diffusion through their gills. So they actually have a special salt gland, which is shown here, and that essentially allows them to concentrate sodium chloride, and then that sodium chloride joins with the urine, and it's excreted from the body. So they have a very interesting, just to recap, they have a very interesting strategy, which is they're actually hyperosmotic relative to seawater because of the high level of urea and trimethylamine oxide. So water enters, they get rid of the excess water through the urine, and then they do get rid of salts that are in excess using a special salt gland. Let's now look at freshwater animals, and we'll use fish as our example. So the tissue of freshwater animals is hyperosmotic relative to the environment. So they're living in a lake or a river, which has really a very low salt concentration. So for this reason, the water concentration of their environment is higher than their tissue concentration. So they're going to constantly take in water by osmosis from their hypoosmotic environment. And their tissues are also salt through the environment. So they're going to lose salts by diffusion. So they're going to have to maintain water balance by drinking almost no water. Remember, they're going to be taking in water all the time, right, through osmosis. So they don't want to really add to that by drinking water. And so water is entering their body, so they're going to need to get rid of that water. So they're going to excrete large or copious amounts of dilute urine. A dilute urine because they're trying to hold on to the salts that they have in their body. And okay, so salts are lost by diffusion, and those salts that are lost by diffusion across the surface of the body are going to be replaced by salts in food. And they also have special uptake mechanisms. So these are active transport mechanisms, proteins that require energy in the form of ATP to bring ions such as sodium and chloride back into the body. Here's a diagram from your book showing a perch. So this is a freshwater fish. Again, the gills are going to be sort of the vulnerable point. There's a large surface area, they're delicate structures. So there's a relatively easy way for water to get through from the environment and into the blood plasma there. So because they're more concentrated than the environment, water will enter via osmosis into their blood plasma through the gills. So they're going to essentially have this influx of water. To get rid of that, they're going to create a lot of urine, and this urine will be relatively dilute. It won't have a lot of salts in it. Now, because they're saltier than the environment, they're losing salts all the time through urine and other across body surfaces. So they're going to need to take up salt from the environment, and they do that again across the gill surfaces. There are specific transport proteins that will bring sodium, chloride, potassium, and so forth into the blood plasma of this perch. Now, they're also, as they eat, it's almost impossible not to bring in some water. So that's being shown here. They're going to gain water, and they're also going to gain some salts. Here's a comparison of the two types of fish we looked at. We looked at the cod. Recall that it's going to have osmotic loss of water to its very salt environment. So it's just like those cucumbers we saw earlier. It's very salt. Has a lot of water compared to its environment. So the water concentration is low in the environment. Water's moving out, and it's in a salty environment. So it's going to gain salt from food. It's going to end up, because it's losing water, it needs to gain water by drinking. As it does that, it's going to gain salt that way. And so it's going to have a lot of salt that's going to have to actively excrete across the gill surface. Again, that's going to be something that's going to take energy in the form of ATP. Now, because this animal is losing water all the time, it should make a very concentrated urine. In other words, not a very large volume of urine per day. And it should have quite a few salts in it. And then for the freshwater fish, everything's essentially turned around. It's the opposite way. So they're going to have osmotic water gain through the gills, and some water gain through eating. But it would have excess water, so it needs to get rid of it. So it's going to make very copious or large amounts of urine, and this urine will be dilute because essentially this fish needs to retain its salts. In fact, it's going to do active uptake of salts across the gill surfaces, and of course it'll have salts coming in through the food also. So if you imagine an animal like the salmon, which we discussed earlier, it's euryhaline, meaning because it migrates between fresh and seawater. So it's going to actually be able to change the way it osmoregulates between these two systems. So some animals are able to live in a really extreme, harsh environment. So for example, there are aquatic invertebrates that live in temporary ponds, such as the tardigrade, which is also called the water bear. So they're able to essentially go from an aqueous environment into one where there's almost no water, and they can become completely dehydrated. So this animal is not dead, it's just dehydrated. And then when water is applied to it, it can come back to life. So this adaptation is called anhydrobiosis. So a tardigrade can dehydrate from about 85% water in its body to only 2% water in its dehydrated inactive state. So land animals also have problems with water balance. So for example, being on land, by definition, you're not in water. And so one of the biggest problems land animals have is that they have to conserve water because they have water loss all the time. So for example, uh, in humans, one way we minimize water loss is we have an outer dead layer of keratinized cells, which is shown here, and that protects us. That helps keep us from drying out. Um, Many desert animals are nocturnal, which reduces evaporation uh, of water from their various body surfaces. Despite these adaptations, most land animals do lose water. So you're going to lose water in urine, in feces, across the skin through sweating and other things like that. What else? Across respiratory surfaces. So when you breathe, the air in your lungs is essentially at 100% relative humidity for 37 degrees centigrade. And so you lose quite a bit of water just by breathing. To maintain water balance, and animals drink water, they're going to eat moist food that's going to have some water content. They're going to produce water through aerobic respiration, which I mentioned earlier. Not surprisingly, osmoregulation has a cost. So osmoregulators must expend energy to maintain osmotic gradients. And the amount of energy differs based on a couple of different factors. First of all, one factor is how different the animal's osmolarity is from its surroundings. If you have a fish, for example, that has a very different osmolarity from its surroundings, then it's going to expend more energy. So you'll find, for example, if you look at animals that, for example, bony fish that live in the ocean, they're generally going to have slightly higher um, tissue osmolarity compared to fish that are adapted for, say, fresh water. What else? So we can also take some other examples. We could look at, for example, at how easily water and solutes move across an animal surface. So one thing that animals do is they simply make their surfaces less permeable. So for example, cetaceans, which are things like dolphins and whales, they have relatively waterproof keratinized skin. So they don't have a lot of water flow by osmosis through their skin. And of course, they have lungs, not gills, so they come up to the surface to breathe. And so they get around this whole problem of living in the very salty ocean by being essentially waterproof. And they're certainly going to have some areas where they're going to have some osmotic movement of water, but it's limited. And so that limits their energy expenditure. For say a bony fish, the osmoregulation is going to account for about 5% of the resting metabolic rate. So it's pretty significant for them. So another way we can do this, as I mentioned, is how different the animal's osmolarity is from its surroundings. So if we take the example of a mollusk, like a bivalve, these animals are filter feeders. So for example, mussel filters essentially sucks water in, and then it has the water go through its body, and as it does that, it finds plankton and so forth, things in the water to eat. So it has a lot of exposure to its environment. And so it's, if you look at mussels, if you look at a marine mussel, its tissues will have a very high osmolarity. So for example, marine mollusks will have a tissue osmolarity that's about the same as seawater, at 1,000 milliosmoles per liter. For freshwater mollusk, the tissue osmolarity is much lower. It's more in the range of about 40 milliosmoles per liter. And so that's going to reduce the energy expenditure. Freshwater has an osmolarity of about 0.5 to 15 milliosmoles per liter, by way of comparison. So I mentioned earlier the idea that your cells are in a nice warm pond in a sense. They're in an environment where the temperature is correct, the solute concentration is correct, the water concentration is correct. And that allows your cells to do what they're supposed to do. They can go about their business and do their job. And so the overriding concept here is one of what's called homeostasis. So in order for your cells to work properly, the environment that they're in, which is this internal environment, should be relatively stable. So it can be stable in terms of things like potassium concentration, chloride concentration, temperature, pH, and so forth, overall osmotic osmolarity, and so forth. And so this is the idea of homeostasis, that despite variations in the environment, your internal environment is relatively constant. So that's part of what the idea of homeostasis is. It's actually a little more complicated. The other part of homeostasis as a concept is that it's not passive. It's based on having this active system within your body that counteracts these changes in sensed internal variables. So for example, your body will sense body temperature, core body temperature. If it drops too low, then your body will enact certain changes that are designed to maintain the body temperature or bring it back up. If your core body temperature becomes too high, then you'll start sweating and doing other things to sort of get rid of the excess heat. And so the idea is that you have these regulated variables that are sensed by your body, and these regulated variables have set points, and so your body tries to maintain these variables within a certain range. So again, if we use the example of body temperature, your core body temperature should be about 30 degrees Celsius, 37 degrees Celsius. And so any deviations from that value lead to a response to bring the body temperature back into the normal range. And so one of the major targets of homeostasis and these homeostatic mechanisms is the fluid within your body. So for example, your body pays a lot of attention to your blood plasma. It'll pay attention to things like the osmolarity of your blood plasma. It'll pay attention to concentrations of certain ions in your blood plasma also. So now that we've established the idea that we have active mechanisms, homeostatic mechanisms that try to maintain this internal environment with it relatively constant, how do we actually go about that? Well, we've already talked a little bit about this, but we have these active processes that will do things like excretion of salts from the body. And these occur across a special kind of epithelia called transport epithelia. So transport epithelia are involved in osmoregulation, and they're epithelial cells that are specialized for the control of movement of solutes in specific directions. So for example, they might be involved in a fish in the excretion of sodium across the gills of a fish. They're typically can be arranged in complex tubular networks. So for example, in us, we have transport epithelia within our kidneys and the walls of the kidneys. So that would be one example. Another example, which we'll go into some detail right now, is the nasal glands of marine birds. What these are designed to do is remove excess sodium chloride from the blood. As I mentioned at the very beginning of this lecture, the wandering albatross lives day and night on the ocean, only coming to land to reproduce. And so they're drinking seawater, they're eating fish that have very salty flesh. And so it's really a mystery how they're able to do this. I mean, a human would never be able to do that. We simply don't have the ability to osmoregulate to that level. So researchers studied this by taking captive seabirds, and they would do something like, first, a typical experiment go like this. They'd give them fresh water to drink. Then they'd switch their drink to seawater. And then they test the bird's urine, and the urine wouldn't actually be much saltier than it was originally. But so that means the kidneys were not actually excreting this excess salt in the seawater. So then what they found, though, was that there was fluid dripping from the tip of the beaks, and if you measured that, that would that was very concentrated sodium chloride solution. So they could tell then that the excess salt was being excreted through a gland, and the gland is called the nasal salt gland, and you can see it here in the picture. So these glands also are going to use active transport, so they're going to be moving sodium and chloride up their concentration gradient. This is a detailed diagram from your picture showing how these salt glands work. So as I mentioned earlier, some of these transport epithelia are actually arranged into systems of tubes, and that's certainly the case here. So if we look at the nasal salt gland and we zoom in at a part of it, we can see that there's the gland itself. You can see that there's the transport epithelial tissue here, and fluid will move out of the gland into a central duct and finally will exit the animal, so will be excreted. And you'll notice these secretory tubules are very closely juxtaposed to capillaries. So now if we zoom in a section of this, we can see here's blood flowing in one direction. And within the lumen of the secretory fluid, there's a salty solution that's moving in the opposite direction. And what you can see here is that there are going to be active transport mechanisms. They're going to move salt, such as sodium and chloride, from the blood plasma and into the lumen of the secretory tubule. And you can see as the fluid moves along, it starts off not very salty, but it becomes more and more salty as it picks up more and more sodium chloride. And eventually you have an extremely concentrated sodium chloride solution that leaves the nostril. And so this is an example of active transport. It's in which ions are moving uphill against their concentration gradient. And in doing this, this bird is essentially getting rid of the excess sodium and chloride that was in its diet and in whatever water it was drinking, if it drank any seawater. So we're going to end this lecture on this picture. In addition to birds, there are also sea turtles and marine iguanas that also have salt glands. And we actually did mention another animal, which is the shark. Remember, it had a salt gland that would secrete sodium and chloride into the urine. Here's a sea turtle. It has tear ducts that act as their salt gland. Galapagos marine iguanas get rid of salt ingested during feeding underwater, so they feed on a very salty diet of fish. The white crystals on its head are made of excess salt. Again, it's probably mostly sodium and chloride. There may be some other salts, such as calcium, also mixed in here. And these are made in their salt glands. And you can see that they'll actually even sneeze out this very salty, briny solution. Okay, so we'll end there.